I want to thank you. I want to thank the UCLA Staff Assembly for inviting me to talk. And today's talk is called Testing Strategies to Help Manage the COVID-19 Pandemic. So I am the Section Chief and Director of Clinical Microbiology for the UCLA Health System. And in my laboratory for the health system, we perform all of the COVID-19 testing, including PCR and serology testing. So we're going to talk about all of that today. And I want to leave plenty of time for questions at the end. You can put your questions into the chat. We'll be more than happy to answer them. Or at the very end, you can raise your hand and we can call on you to have you answer them. I think it's really, it's great that you have all tuned in today. Part of the issue with this pandemic is that there's a lot of misinformation. And so I really want to thank you all for making a step forward to getting the best information possible, because really our only way out of this is together, and by us together having all of the best information. So hopefully I can provide some of that to you today. So just as a brief introduction to the UCLA Health System. So our health system has two hospitals, Ronald Reagan Hospital and UCLA Santa Monica Hospital. They're about 2 miles apart and constitute about 900 total inpatient beds. And then we have about 225 outpatient care clinics that's spread over 300 square miles of Southern California. We have one centralized infectious disease diagnostic laboratory. So the Clinical Microbiology Laboratory does about 1 million to 1.5 million tests per year. I have 120 staff in the building, and we have multiple areas of microbiology testing. So we do infectious disease testing for bacteriology, for virology, for mycology, which is relevant yeasts and molds, immunoserology and parasitology. From a sample type perspective, you imagine you show up to the doctor, a sample is submitted like blood or respiratory. This makes its way to my laboratory, and then we do a series of tests to assist in the diagnosis of disease. So these diseases include pneumonia, which we're going to talk a lot about today around COVID-19. Also includes things like sepsis, bloodstream infections, urinary tract infections, all of the STD testing, and really any other disease of infectious origin. So taking a closer look at my virology lab specifically, I have maybe between 20 to 30 people that work in the virology laboratory, and the diseases we're helping to diagnose in that setting include herpes, VZV, which is a virus that causes chickenpox, flu A and flu B, and then importantly, COVID-19, which we're going to focus in on today. So let's take a step back and look at a brief history and try and all remember the last, well, it's only been 6 months, but it's felt like the last 10 years of COVID-19 in our lives. So on February 26th, University of California, Davis actually reported the first patient with community-acquired COVID-19. So this was COVID-19 that was acquired within the setting of around Davis itself and not associated with a particular traveler. As of March 13th, so a couple of weeks later, we diagnosed our first patient at UCLA, which is at UCLA Santa Monica Hospital. 4 days later, the hospital became closed entirely to all outside visitors. And 2 days after that, we had our stay-at-home order in California. So some of our challenges within that first week at our hospitals were really around testing. At that time, we didn't have enough COVID-19 tests to perform, so we had to ration those tests. And we're going to talk in detail today as to why that sort of thing happened. And then there were some questions on sensitivity, i.e., is the test giving a false negative when it gives a result back? We're going to talk in a lot of detail about that because that's still a question that's out in the community. At that same time, we were trying to figure out from a hospital side how to contain patients and not let disease spread around the hospital. And this is this concept of enhanced droplet isolation. Of course, there were lots of potential treatment options that were being considered by our treating clinicians. And then if you remember at that same time, all of the really scary reports that were coming out of New York City about ventilator shortages, about hospital-acquired infections. So it was really a time with a lot of unknowns. I want to discuss the disease specifically. And so there's a certain confusion around the names for this particular disease, right? Some people say COVID-19, some people say SARS-CoV-2. And so in breaking that down, COVID-19 is actually an acronym. The acronym stands for Corona, the CO, virus, the VI, disease, 2019. And the 19 comes from the year that it came about as a virus that we understand in causing disease. So that's COVID-19. So when people talk about COVID-19, they're talking about the disease itself. The virus that causes COVID-19 is called SARS-CoV-2, and we'll talk about the origin of that. You could think of this in the same way that HIV is a virus that causes AIDS. SARS-CoV-2 is a virus that causes COVID-19. So when we talk about COVID-19, we're talking about the disease. So then what does this mean from catching this particular infection? So when a person encounters the SARS-CoV-2 virus, there is a certain incubation period where that virus isn't showing any symptoms in that particular person. And this can be anywhere from 0 to 14 days. Now, typically the average is 5 days. 5 days after exposure, a person will become symptomatic if they are going to become symptomatic. And so you see in this incubation period, you have this countdown to the onset of symptoms. It's understood now that about 2 days before symptom onset occurs, a person can be contagious with the virus, i.e. have enough virus to be able to pass the virus along to another person. So in that setting, that's what we call the pre-symptomatic window. Then after the onset of symptoms, the symptoms that we look for are fever, cough, fatigue, and then myalgias, body aches, potentially diarrhea, potentially some upper respiratory tract infection symptoms like runny nose. All of these things can happen at the onset of symptoms. And then over the course of time, what you'll see is that 6 to 7 days later, from a person having symptoms, 40% of symptomatic patients will have dyspnea. This is a trouble breathing. And then a small subset of those patients can move on to critical illness. So 5% in symptomatic, and then potentially severe illness. And these are what require hospitalization. So if you take the large group of people that are infected, we don't know how many people actually move on to being symptomatic, but we do know of that symptomatic group, only 60 to 70% of them should have what's considered to be mild disease, whereas 30 to 40% can have increased disease, and that of course correlates with comorbidities. Right? People who are older, people who potentially have comorbidities associated with worse infectious disease, let's say people who are immune compromised, can move into this area of critical or severe illness. Now, COVID-19 isn't the only infectious disease that can cause pneumonia, right? Pneumonia, which is an infection of the lower respiratory tract of the lungs, can be caused by bacteria, can be caused by different fungus, and can be caused by a series of viruses. This is just a list of all of the viruses that potentially can cause pneumonia. But in the immune competent person, this is the person who has a active immune system, we really only think of a couple of different viruses that can cause viral pneumonia. One, of course, is flu, right? Every season, flu comes around, and ultimately people that are healthy and immune competent can get viral pneumonia. A lot of these other viruses that are on here usually just cause the common cold in people who have an immune system, who have a healthy immune system, but can cause severe pneumonia in patients that are immune compromised. One of those groups of viruses that historically we only think of as causing the common cold are this group of viruses called the coronaviruses, of which COVID-19 or SARS-CoV-2, COVID-19 disease, SARS-CoV-2, the virus, is actually a part of. So I want to walk you through this group of viruses, a very fascinating group of viruses. So these are enveloped, non-segmented, single-stranded, positive sense RNA viruses. All of that means that sort of in their structure, they're very similar to a lot of other cold viruses. And they're named corona because on electron micrograph, there's a spike protein. This is the protein that actually facilitates the virus's ability to bind and infect a cell, forms this really pretty little crown around the organism. So that's where it gets this name of corona. Now, coronaviruses are known animal viruses. This is going to become important as we think about the spread of COVID-19. So they were recognized as animal pathogens in the 30s, and then it wasn't really recognized until 30 years later that these viruses could cause disease in human beings. Now, in studying animals, what we find is that they either cause respiratory illness or GI illness, and rarely any other kinds of disseminated disease in animals. This is a table of the known human coronaviruses. These circle every year, they circle the globe, they cause a cold, and ultimately they affect upper respiratory tract or cause cold-like viral symptoms. So this ties in well to this concept called spillover, and I want to give a shout out to one of my favorite books by David Quammen called Spillover, and it tracks the movement of pathogens from an animal species into a human being. And so these are infections that are called zoonotic. A lot of infections that humans experience are actually zoonotic diseases, and coronaviruses all fit into this category. So if we look at some of the human coronaviruses, right, these viruses that we know cause colds in human beings now, these viruses all have an animal origin. So if you look at NL63 is a human coronavirus that started in a bat. There was usually some level of intermediate host that brought that virus closer to people. There isn't a lot of contact between people and bats. And so if you look at something like OC43, the human coronavirus, it started in a rat. That rat then spread that virus to domestic cattle, and then domestic cattle spread that virus to human beings, and there was human-to-human transmission. I say all this to a little bit break down the sort of mystery around COVID-19. I think there's a lot of discussion on, oh, this moved over from an animal and it's never happened before, and why are we experiencing this? This happens all the time with coronaviruses. This is actually the pathway that coronaviruses take to causing human disease. And what we learned from more serious coronaviruses, which includes the SARS outbreak in 2003 and the ongoing MERS outbreak, is that even though many coronavirus species can just cause colds in human beings, some of them can cause worse respiratory illness. So, I think a good question then comes up. How does a spillover, which is this event where an animal virus or an animal pathogen makes one person sick, become a pandemic, which is a disease that spreads around the globe? So the way this happens is you have a virus, and the virus has high host plasticity. What this means is that the virus can infect lots of different species. And the way that it does this is with a shared receptor. So the receptor that's used for COVID-19 to infect human beings, called ACE2, is also found on other animals. So it can cause respiratory diseases in other animals. As human beings come into contact with animals, and we all know if you're a dog or cat lover, you come into contact with an animal every single day. Don't think you can't get diseases from your pets. Or let's say you work in domestic farming, or you work with wild animals, there are going to be potential diseases that can pass back and forth. So that is spillover to a human. Now, most spillover events don't then lead to human-to-human transmission, meaning that pathogen or that virus was competent to infect that one person, but couldn't actually infect other people from that infected person. That's a much more rare event, but when that does happen, when you have human-to-human transmission, that's when you get amplification that can spread around the globe. As we all know, we can get on an airplane, or at least pre-COVID, we could get on an airplane, and you could be on the other side of the world in 14 hours. This allows for global spread of the virus. And so in thinking about COVID-19, this is exactly what happened. This is also what happened in the 30s with an animal virus that causes the human coronavirus that ends up just causing a cold. So again, this global transmission is something that can happen within that setting. It's just we haven't seen it happen on this scale. So if you are following along the pandemic, this happens to be my favorite dashboard. It's from Johns Hopkins. And what this shows as of yesterday at 10:26 a.m., we were close to 30 million cases globally, almost a million deaths globally. And this is a really nice tracker for the global information for this particular pandemic. And of course, we all know with this pandemic, the United States is one of the hardest hit countries. And so if you look at cases by US county, you can see that Los Angeles is unfortunately leading this with a total of 254,000 confirmed cases. But I think what's interesting about this map is there's really no place in the United States that COVID-19 hasn't touched. And whether it's in the major cities like New York or Chicago, or smaller cities like I'm from Racine, Wisconsin, you can have outbreaks in all of these different areas across the country. So how did we get here, and how is testing involved in this? So I'm going to tell a little bit of a backstory around COVID-19, especially focusing in on testing, and then talk specifically about what happens for testing for this particular virus. So if you remember back to December 2019, when COVID-19 started, there was a cluster of pneumonia cases, so lower respiratory tract cases in China of unknown cause. So this means these patients were tested for all the known things. If you'll remember that list that I showed, things like influenza, RSV, they tested them for SARS, they tested them from all of these things, and they were negative, but they had pneumonia. And it appeared that that pneumonia was even human-to-human transmissible. And so there was some worry about a new pathogen, right? Pretty quickly after that, about a week and a half after that, the Chinese CDC actually identified a novel coronavirus, and this novel coronavirus was whole genome sequenced. What happens there is you actually look at all of the RNA that's associated with this particular virus, and what they found was that it was a very close cousin to the original SARS, which was the severe acute respiratory syndrome virus that caused the outbreak in 2003. It was so closely related that they named it SARS-CoV-2. And SARS-CoV is the original virus. So this is where that name came from. All of the cases were thought to be linked to a single food market. And this food market, so there's a representative picture here, of course, is full of a lot of people, but also has wild animals mixing with domestic animals. And so, I think that ultimately food markets like this represent a space where spillover can occur. But I want to emphasize most cultures across the world have these types of markets, right? And so these types of events can possibly happen. So what did we do then at UCLA with the recognition of this new particular virus? So what happened is that by January 15th, UCLA was actually screening people for persons under investigation. At this time, it meant people travel history to mainland China who were showing up with symptoms of cough, shortness of breath, and fever, because at that time, it was thought that those were the only symptoms associated with COVID-19. Now, UCLA is one of the screening hospitals for LAX, so if somebody shows up at the airport sick, they get transported to the Ronald Reagan Medical Center. So we needed to be able to care for these particular patients. An important part for caring these patients is figuring out whether they have COVID-19 at all, right? A person can show up with symptoms, and the symptoms of the flu are very similar to the symptoms of COVID-19. You can't just look at a patient and diagnose the disease. So this is where testing is very, very important. The challenge at this time is, you'll remember I introduced you to that entire large hospital lab, my lab, that can do testing. As of January, we weren't allowed to do COVID-19 testing at my hospital. In fact, the only place you could get a COVID-19 test was at the CDC in Atlanta. Now, everyone on this call will know Atlanta is nowhere near Los Angeles. So ultimately, attempting to get a PCR result, which is the type of test you perform for COVID-19, for your rule out was taking 5 to 6 days. So in that setting, it made it very difficult to get on top of a new infection because you couldn't test people very readily. Everyone recognized that this was a problem, and the CDC quickly became overwhelmed with requests to do this type of testing. And so the CDC in February decided to push out tests to the state and local public health laboratories. Los Angeles County has 10 million people. It's got more people than a lot of states have. So we actually have one of the premier public health laboratories in the entire country. They were going to be one of the first to get this test for COVID-19. Unfortunately, the test that got pushed out by the CDC had a problem with it. So we're going to talk a little bit about the targets, but there were 3 viral gene targets in the test, and all 3 had to be positive for you to correctly call COVID-19. But one of the targets wasn't working correctly. So it really delayed our public health lab's ability to bring on the test. So by the time they brought on the test in mid to late February, we had too many cases potentially in Los Angeles, and it slowed down the turnaround time. So we would send from UCLA now to Downey, where our public health lab is, but it was still taking more than a day to get the result back. So we weren't able to test as many people as we possibly could. This brought us to the end of February. Now, again, remember, this started at the end of December, and now we're already at the end of February. And that's when the CDC and the FDA decided that my laboratory, so a hospital-based laboratory, could perform testing for COVID-19. The challenge is they did not make it easy. So what they said was that you could perform just the CDC test, that same test that was offered at the CDC and the state public health labs. But in order to do that, you needed specialized equipment that wasn't available in most clinical labs. You needed highly trained technologists to be able to do that testing. And so ultimately across the country, most labs couldn't bring up the test. So they released a test that very, very few labs outside of academic medical centers could actually perform. They had another pathway of which they told labs you could make your own test, and even fewer labs were able to do that. So ultimately what this meant was even by early March, there were very few places within the United States that you could actually get testing for what we know now was a rapidly growing disease across the United States. So let's talk about the testing specifically. How does this work? Because I think there's misunderstanding out there how testing is performed. And so testing starts at sample collection. If anybody on this call has been tested for COVID-19, you'll be very familiar with this process. So you show up at what's called a testing site. Now, unfortunately, this is a bit of a misnomer because no testing actually happens at these sites. This is actually a collection site. So what happens at the collection site, be it a hospital or a doctor's office, or if you're with us in LA, potentially Dodger Stadium, is that a sample is collected, and typically that sample was a nasopharyngeal swab. Here you can kind of see how deep that swab goes back into the nasal cavity. The question is why? Why does it need to go back so deep? Why do you have to take that type of specimen? Ultimately, this place back here in the nasopharyngeal region where the nasal cavity meets the oropharyngeal cavity is where viruses love to live. And so ultimately, that's also the best place to test for flu and a lot of other viral pathogens. So you go way back there to collect that and put it into a transport vial. Now, there are other collection schemes that are out there now, including saliva, anterior nares, so just at the end of the nose, throat swab. More than happy to answer any questions about the differences between those sample types. But ultimately, once the sample collected, it's just the first start of the journey. The next thing that has to happen is that that sample needs to move to a centralized laboratory, a laboratory like mine at UCLA. Now, this can lead to significant delays. So for example, at UCLA, the average time from collection to getting to my laboratory is about 4 to 6 hours, relatively fast. There are some states, and I mean actually full states, that don't even have testing facilities in that state. And so what that means is that that sample may need to travel for 2 or 3 days to actually get to a centralized testing facility to be performed. So this is one reason why across the country there are places that people are waiting for 7 to 14 days to actually have a test result. Then once it arrives in the centralized laboratory, how fast it gets tested depends on the queue in that laboratory. So for example, if you have a laboratory that can test, let's say 10,000 samples a day, but there's a queue of 50,000 samples waiting to be tested, you now have a 5-day delay for anything showing up in that door to then be tested. Functionally at UCLA, we clear our test results every single day, i.e. we get everything back within 24 hours. So that's another reason why delay can occur. And remember, we haven't even started testing yet. So now, once the testing process begins, it's really a 2-step process. The first step is called DNA RNA extraction. You take that sample that has all the cells and all the fluid and maybe virus in it, and you extract all of the DNA and RNA out of that sample. So what you end up with is a purified set of DNA and RNA. And then you run a test called a reverse transcriptase PCR. What this test does is very sensitively looks for the presence of viral RNA in that material. If it's there, detect it. If it's not there, not detect it. So ultimately, that's the entire process that needs to happen for a test result to be returned. This is a schema of the virus. So the virus is composed of different proteins. Here's that spike protein that I showed you before that the coronavirus is named for. And there are different proteins on the inside, and then there's the genomic RNA. The PCR tests actually target different parts of the genomic RNA. So if you lay out the genome here, there are different genomic segments, and then you can look for viral RNA pieces that are specific to that particular virus. Now, this is important because you don't want cross-reactivity, right? If I targeted an area of that viral genome that, let's say, looked a whole lot like the flu. So in that RNA genome segment, the flu and coronavirus are very similar. If I target that by my experiment, then ultimately I won't know if it's the difference between flu and something like COVID-19. So you look for areas of the genome that are specific to SARS-CoV-2. I say all that because we're about to walk our way through the tests that are available in my lab, because I actually have more than one PCR test for COVID-19. So the first test I have is the CDC test. If you remember, this was the original test that was made. When we brought this on in early March, this test uses 2 different targets on the nucleocapsid gene. And the challenge of this test, remember at this time, this was the only test available across the country. It takes about 8 total hours to do 30 patients. Just to give you a clue, right now, I do about 1,600 patients a day in my laboratory. So at this point, I could only do 30. Again, part of the challenge of expanding testing was that this was not a high throughput test. So this is a test we really only reserve for our sickest patients, because we can run lower respiratory tract specimens, not the nasopharyngeal swab, but stuff from deep in the lung from very, very sick patients. We were waiting for the FDA to approve faster test methods, and this is just a table representing by date the types of tests that became available. So here you see February 4th, the CDC test was approved. It wasn't released until end of February, and then other manufacturers started to make tests. Here is the test we moved forward with. It's called the Diasorin molecular test. To introduce you to this test, it's a PCR test. 2 different targets on the virus. The great thing about this test, though, is it only takes 90 minutes to perform. Remember, CDC test, 8 hours. This test, only 90 minutes to perform. And what that allows us to do when we brought this on then is to run it at a much higher scale. We're able to run this test at about 300 tests per day. And we were able to turn around. So turnaround time is the time from collection to result, right? That whole thing that I laid out before, in 4 to 6 hours for our inpatients. This was critical because inpatient medicine depends on moving and treating COVID-19 patients appropriately, so you don't have the virus spreading throughout your hospital. So by March 21st, we were able to bring this test on, and we're offering a 4 to 6 hour turnaround time for our inpatient testing results. The challenge still, though, is I could only perform 330 tests a day. Again, I needed to be up around 1,600 tests a day to support our UCLA health population. So I now needed to bring in a 3rd PCR test to perform for COVID-19. So this was the 3rd platform we looked at. It's called the Thermo Fisher platform. Relatively manual. It has a couple of instruments. The key to this, though, is that you can expand it up dramatically if you have the expertise in your laboratory. So we were able to actually run this test at about 1,000 samples per day. And so if you combine that with the faster Diasorin test, with the CDC test, we now are able to have a total amount of PCR testing that can support potentially our needs of our health system. This test takes longer, though. So ultimately, from a turnaround time perspective, you can see we were mapping this in April. And in that mapping, it was anywhere between 10 to 20 hours as a turnaround time. So we really use this as an outpatient test. So if somebody shows up in our ambulatory clinic or somebody shows up to one of our drive-thrus, we get the test result either same day or next day, and that's appropriate for patient care. I want to emphasize, we are unusual at UCLA. Even now, most places aren't able to offer this high level of service for testing. And even now, a lot of places, people are waiting 7 to 14 days for a test result. That really hampers symptomatic testing for this particular disease and makes it nearly impossible to understand how many people have the virus and to be able to mitigate the spread of the virus. So I would like to say that we're better off overall in this. There are tests that are available, but it's very, very difficult to be able to get your hands on these, especially from a centralized laboratory perspective. So, I think one of the natural questions that comes up is, I've just told you we have 3 different tests for the exact same virus. You don't want to get the bad tests, right? How do I know all 3 of these tests actually perform exactly the same? Which is critically important if I'm not going to use all 3 on all 3 patient populations. And so we needed to study that. And so in this setting, we took our first 10,000 test results. At this point, we've actually done 100,000 tests, PCR tests, but we took our first 10,000 and we compared the 3 assays. The CDC assay, the original one, the Simplexa, which is the fast one for our inpatient population, and this TACPath assay for our outpatient. And what we found across the board is that they behaved very similarly. The positivity rates were about the same. This thing called cycle threshold, because you have an idea how much virus is there, was very similar across the board. And we didn't see a lot of either invalid or inconclusive results. We'll get into what those mean a little bit later, but what this suggested to us is we could continue down this pathway, having these 3 different tests and have the equivalent sensitivity and specificity, meaning trustworthiness of the results, no matter which one of the tests were performed. This is really important as well when you think about, maybe you've heard of antigen testing. So there are some new tests that are on the market that are much more rapid. Those tests actually suffer in some of the concepts of releasing a false negative versus a true negative. And so we wanted to be sure that wasn't happening with our particular test. So then, and I take this question a lot from our clinicians and of course from friends and family, is this test sensitive? And sensitivity is really this measure of how sure are you if the test says it's negative that the patient doesn't have COVID-19? We need to be sure of that, right? And there's a couple of different parameters that go into that. One is this concept of analytical sensitivity. And this is a question is how good is the PCR at finding that viral RNA? And what we find is that the PCR is very good. So it gets down to about 100 copies of virus per mil, which is about as sensitive as it gets. So in that setting, what I tell our clinicians is if the virus is in the sample, we'll find it. But the question really is broader than that, right? Is the virus in the sample? And this is actually a complicated question, right? One, it depends on how good the collection was. Did we do the nasopharyngeal or did we just do a throat swab? Because in that setting, some specimens have a higher sensitivity, meaning likelihood to have the virus, than other specimens. And again, the other challenge with this particular disease, and remember, we're only 7 or 8 months into even understanding this disease, it appears to move around. So it may start in the nasopharyngeal area, but then as it becomes more severe, it may move into the lower lungs. And we really don't understand the dynamics in the asymptomatic patient or in that pre-symptomatic window, how much virus a person has. So all of that is going to be dependent on whether or not we release a false negative or a true negative. So how do we look at this to determine how good our test was? So the way that we look at this is by repeat testing. And so what I mean is that a patient shows up, gets a nasopharyngeal swab, and we release a negative result. And then for whatever reason, they're tested again. In the setting of a person being tested again, if the first one is negative and the second one is negative, true negative. But if the first one is negative and the second one is positive, we're arguing that first one was a false negative, right? So in that setting, that's our real measure of clinical sensitivity. And what we find is that 98% of the time, if the first one is negative, any subsequent one tested will also be negative. That's what this data says down here, and it says it for any of the tests that we perform. So we have a 98% confidence that if you get a negative from us, you don't have COVID-19. This is the equivalent, nothing is 100%. There is no test that's just perfect. This is the best as it gets in infectious disease diagnostics. So then the next question that has come up in the field is, well, what specimen is best, right? Should we have a nasopharyngeal swab specimen, and especially on the inpatient side, or should we have something lower respiratory like sputum? BAL is a bronchial alveolar lavage. It's like a wash in the lower lungs that takes specimen. For our very, very sick population, what's going to be best? So one way we look at that is by looking at the overall positivity rates. And what we find is that 7% of our BAL is positive, but only 3% of our nasopharyngeal swabs. Way less numbers tested. But does this say a BAL is better for very sick patients? And what we found is that all of our very, very sick patients, so these 20 patients that have been positive, not only have had positive BAL, but they've also had a positive nasopharyngeal swab. So what it says to us in our population for our people who are hospitalized is that both is probably the best way to go about testing, but we haven't had somebody who's lower respiratory tract negative, upper respiratory tract positive. So another question that comes up is this concept of an inconclusive result. And so, there are actually 3 different results you can get from a COVID-19 test. Positive, you have COVID-19. Negative, you don't have COVID-19. Or inconclusive, which is, we don't know, you might have it, you might not. So in that setting, there are a couple of different ways that this happens. I'll let you know our inconclusive rate is around 0.5% for the tests that we're running. So it's a really, really low number of patients that actually get an inconclusive result. But we wanted to do a look back to see whether or not those results in further testing were true positives or they were actually negatives. And what we find is that it really depends on the symptoms of the patient. So these ones in green that ended up being true positives after inconclusives, these were people who became symptomatic later. And as they became symptomatic, we found out that the original result, that inconclusive, was a very weak positive. But then we had some patients that weren't symptomatic to be started. Let's say they were tested for asymptomatic screening for surgery. Those patients on an inconclusive on follow-up testing ended up being negative. And so our suggestion now, and our clinicians will do this, is if you get an inconclusive result, get tested again. And when you get tested again, the next result will be definitive, either positive or negative. So this leads us to our current dashboard. So what you can see at this point at UCLA, in my microbiology laboratory, we have tested about 79,000 patients. And of those patients tested, 75,000 have been negative. Total positive patients were about 3,000 total. And currently we are at our lowest ever hospitalization rate with only 14 patients hospitalized with COVID-19. What you can see between March 11th and September 12th is that as we grew in our ability to do more and more testing, we saw an early peak, and the red bar here are positive patients. So an early peak in positive patients that came back down. And then if you all remember in Los Angeles, we had another peak in early to mid-July. You can see that here, the peak coming up in early mid-July of positive patients that have then come back down. So overall, what we're finding from a testing perspective is we're still doing a lot of testing, but we are finding far fewer patients as represented by the population at UCLA Health, which is mostly the population that is on the west side of Los Angeles. This is also mimicked by our inpatient numbers seen during the day. So this is a graph from the entire UC system. So this includes UCLA Health Centers, UC Health Centers of UCSF, UCSD, UCLA, UC Irvine, and UC Davis. And what you can see overall is that the numbers peaked in July and our inpatient numbers have been going back down. And you can see that that graph is also happening at the other institutions. But ultimately, UCLA Health is serving a lot of patients that are very, very sick with COVID-19. So that's acute testing. All of the PCR testing is really this question of, do I have acute disease? But there's a second question that's being asked out there from testing, which is, have I had the disease? And if I have had it, am I protected from getting it again? And this is this concept of serology. So in the last kind of 5 to 10 minutes, I want to switch over and talk about serological testing. To put us all on the same page of what we mean, serology is the measurement of these down here. They're called antibodies. These are very specific proteins that are made by your immune system to fight disease. But it takes a while for your immune system to make them. So here, this sort of green, red spiked guy, we'll call that our coronavirus. Coronavirus gets into the system, and what happens is that a macrophage or antigen presenting cell will pick up that virus. It'll recognize it as foreign, and maybe this is happening in the lung or the upper respiratory tract area, and it'll present that to your immune system. You have these cells called T cells and B cells that participate in your specific immune response. Once that pathogen is specifically recognized, there will be an activated B cell that will make antibodies, and those antibodies will bind specifically to that pathogen. It's that response that we look for to see whether or not somebody has been exposed to the disease, and it's that response that ideally leads to immunity. Because once you have these circulating antibodies, if this pathogen gets in again, it will be quickly cleared. The immune system, the antibody response we look for is called IgG, and this is really the convalescent response after you've had an infection. So lots of unknowns here. First, it's unclear if antibodies are protective or neutralizing, meaning the antibodies that we measure, we don't know whether or not they stop a person from getting the disease again. We're hopeful. There's some data to suggest that it will, and ultimately we're hoping that vaccine can produce antibodies like this so people are protected, but right now we don't know. The other thing we're not sure exactly is how often this antibody response occurs and how sick do you need to be to have this happen. So if somebody has very, very mild illness, do they make antibodies? Unknown. If somebody has very, very severe illness, do they make antibodies? It seems like they do. And what about the people who are asymptomatically infected, right? They have the disease, you can test for it, but they never have symptoms. Do they make antibodies? We don't know. But we can say that the average days following infection for antibody production is about 28 days later. Some people show up as early as 14 days later, but it really is a test after disease has happened. So we offer this test at UCLA. Our antibody target, so what antibodies we're looking for are the antibodies that actually bind to that spike protein that we talked about before. It's got pretty good sensitivity and specificity, and we're actually doing some studies now to figure out what population we have at UCLA didn't develop antibodies if they were PCR positive and why. And if a population did develop antibodies, how much antibodies? So there's ongoing research that we're doing for this. But there's a challenge with this type of antibody testing. So specificity is the concept of whether or not you give a false positive. And a false positive is dangerous in the antibody setting, right? Because if I tell you you have antibodies to COVID-19, you may make the decision, you know what, I'm protected. I don't need to wear a mask. I can go to indoor restaurants. I can do all of these things because I can't catch it again and I can't transmit it to other people. We don't know any of that. So right now, it's very difficult to understand what to do with a positive antibody result. In addition, there's a concept called positive predictive value. And this gets a little bit wonky lab medicine. But what this says is that the lower the prevalence of the disease, so the less likely that a person has the disease, the lower the chance that the positive result you get is a true positive versus a false positive. So what that ultimately means is that in areas of the population where the prevalence is 1%, and so that's what I would consider this should be the general community, people who haven't had symptoms or maybe had mild symptoms, only 1% or less of that community is actually going to have experienced COVID-19. A serology test being positive is about 50-50 true positive versus false positive. Well, that's frustrating, right? Because in that setting, then you really can't interpret a positive result. Whereas if you increase that prevalence to 10%, so let's say healthcare workers, frontline responders, people who had symptoms or were part of an outbreak, the prevalence is higher, and now you have what's called a positive predictive value of 90%. That means 9 out of 10 times you can trust that positive result. So all of this we need to take into account in figuring out who should get a serology test and how we even interpret that. So at UCLA, we have pretty strict guidelines. So ultimately, we should only consider even testing someone for serology in the setting of suspected infection that wasn't tested by PCR. So somebody got really, really sick in March, right set of symptoms, never had the PCR test. We can test them for serology later to see if that was COVID-19. Healthcare workers are first responders because, of course, they'll have a higher prevalence. Plasma donation for convalescent plasma treatment. And this is one of the treatments that's being looked at now. So people who have had COVID-19 and have the antibodies, they can donate that plasma because there's some data to suggest that that can be used to treat patients who are sick. But, and this is important, serology testing should never be used to diagnose acute COVID-19. Remember, it takes 21 to 28 days after infection for somebody to even have these antibodies. So if somebody's trying to use one of these tests to figure out whether they have COVID-19 right now, and the test is negative, it doesn't say anything to that person. So ultimately, that person could then go out and potentially spread that disease. So it's not appropriate in that setting in any regard. And also, it's not good to use on the general public. Because again, this is back to this prevalence idea and this notion that we don't know whether it'll be a true positive or a false positive. So right now, serology testing is pretty counterindicated. It's not very useful. And if somebody feels like they have COVID-19 or they've been exposed to COVID-19, it's really the PCR test that's going to be the differential. So how do we get out of this, right? And I took this slide from one of our infectious disease colleagues, one of our infectious disease clinician colleagues, who I recently gave grand rounds to our Department of Medicine. And this is Dr. Vijayan, and her slide was very, very simple. Our way out of this is masking. Now, I know it's been controversial because early on that was not the recommendation, but remember, we're learning things about this virus constantly. And all of the data now says that masking is key to slowing the spread. And so we're able to mask. We can bring down overall the amount of infection. We're not going to test our way out of this problem. right? We can't provide a test to everybody, figure out exactly who's infected, and stop everybody from testing. Because even at a 2% error rate, you know, if you have a 2% false negative rate, but you're testing 100 million people, 2% is a lot of people in 100 million. So again, behavior is what gets us out of this. Education and masking, and then of course a vaccine. And there are some leading candidates, either the Moderna or the AstraZeneca trial, which are both recruiting at UCLA and led by UCLA investigators. The early studies are showing that the immune response to these vaccines are positive. And so I'm optimistic, I'm optimistic that sometime in early 2021, we are going to have vaccines. The vaccines are going to work. Ultimately, the distribution of those vaccines, the equity around the distribution of those vaccines is going to be a very complicated topic. We're not good at healthcare equity in the United States. In fact, I would argue that we're terrible at it. So ultimately, all of that's going to be tied into this process, but it is going to be these two concepts of masking and the vaccine that help us to get out of this. So I want to thank you all for your time. Lab medicine has never really been at the forefront of the discussion of healthcare across the country. And so recently, my laboratory has been featured in the LA Times. So I want to give a shout out to my lab. This is my senior specialist, Brian Bolin, performing testing. Here are other lab members, and they took a picture of me behind my desk. We've really been working overtime to be able to do as much testing as possible. And this is my group in virology. They work overtime nights, they work overtime weekends, just to provide as high a level of testing and patient care as we can to try and fight this pandemic. So really, all thanks to them. I want to thank you all for joining me today, and I believe I've left about 10 minutes for questions.
Thank you so much, Omai. This has been fantastic. We do have a couple of questions that are in the chat. Others, feel free to add questions in the chat or using the raise hand feature if you'd like. I'm going to read one of them from Maria. She asks, the news stated 23andMe stated they found people with blood type O had less cases of COVID-19. Have you found that to be true?
So Maria, that's a great question. I'm just going to be honest with you. I have no idea. And so part of the challenge of COVID-19 is that we've just been overwhelmed with clinical service. And so there hasn't been the opportunity to ask the types of questions that you're asking, right? And so I think that this is where clinical research needs to catch up. And this is why, as we're moving forward with this particular disease, we're learning more and more. And one of the early pieces of data that came out was that it appeared that people with blood type O had less severe disease to COVID-19. Now, is that unusual or possible? Of course it's possible. And there are other blood types that are associated with protection from different types of diseases. And so I think in that setting, these are things we need to explore. These are things we need to figure out. Look, ultimately, even if we are masking and have a vaccine, COVID-19 isn't going anywhere. This is a disease that is now going to be with us. It's this understanding of the disease is now endemic, meaning there are enough people that have been infected that we're not going to be able to eradicate this particular disease. But can we make it like a lot of our circulating viruses, of which don't cause sort of globally 1 million deaths a year, and ultimately back down to the levels that we see on other things? And I think that that's something that still needs to be explored.
Excellent. And another question from AB. Isn't the turnaround too quick for these vaccines to be ready by target date 2021? What are your thoughts?
Yeah, so, and I try and listen to everything that Dr. Fauci puts out there, because ultimately he is our leading expert in infectious disease and leading us through this. And he even emphasized, look, the normal time to make a vaccine is 4 years. It just is. Is it safe then to ramp up vaccine production to make it happen in like a year? And I would argue that with the resources that are thrown behind this, my answer is yes. And all of the companies have come together and said, we are not going to release a product that is neither safe nor effective. And so in that setting, the checkpoints are in place to make sure that the vaccine is safe. Yes, it's ramped up, but a lot of the challenge of making a vaccine and why it takes so long is because you need a certain number of people infected to prove your vaccine works. So if you're making a vaccine for a disease that just there aren't that many infections, you have to wait for that number of people to be infected to prove it in that phase 3 trial. One quote unquote advantage to how poorly the United States is handling COVID-19 is that there are a lot of infected people. And if there are going to be a lot of infected people, you can then accelerate your timeline for being able to prove whether or not your vaccine works, because a large number of people are going to be exposed in the vaccine trials. So I'm hopeful. You know, I think that the companies have made clear they're not going to cut corners, because safety is as big a concern as efficacy. But I am confident that when they are released, they will be safe. And my hope is that they are also efficacious.
Excellent. And another question about those vaccines, Karen asks, what is the difference between these potential vaccines that you mentioned?
Yeah, so that's another great question. There's a lot of ways to make a vaccine. So one way is to sort of take a live virus and beat it up a little bit, and then give that live virus to the system, let the immune system see that virus, but not have it cause a lot of disease, and then you get protection from that. It's the way our influenza virus works. It's the way our measles, mumps, rubella, and VZV viruses work. Another way to make a vaccine is just to take a portion, so a protein from that virus, give that to the immune system. This is how your hepatitis B vaccinations work, and that works as well. A new way, and this is also being explored, is to just use the nucleic acid from that virus to infect this, to be put into a cell to make its own protein, viral protein, and you make it a better immune response. I'm not an expert in vaccines, so I can't tell you which one is better. All I can tell you is that the manufacturers are pursuing all of these lines to be able to figure out which one's going to get the best response.
Great. And then Jeffrey is asking, we've heard about price gouging for SARS-CoV-2 testing. What does UCLA charge, and what is a reasonable charge in the community? So
the charge for testing is complicated. And when I say it's complicated, I mean it's very much tied into insurance companies and Medicare reimbursement. And so just to let you know, the hospital doesn't set the charge specifically. And so for COVID-19 testing, Medicare, which is the largest reimburser in the country, has decided as a group that the reimbursement rate for COVID-19 for a test, or the charge to the patient's insurance, is $100 a test. And so that's the charge that we have, is $100 a test. Now, ultimately, will some places charge more than that? Yes. Will some places charge less than that? That's not usually that common, because ultimately it's very expensive to be able to put together the expert personnel and the machines that are necessary to run this test. Now, I'm going to differentiate that from what I do consider to be price gouging, which is this concept of, if you show up to, let's say, an urgent care in your neighborhood, and they are offering you a COVID test for $200 cash at hand. That is not an appropriate testing scheme, and a lot of times those are serology tests. So people are really sort of paying out of hand for tests that are not appropriate and are not actually giving the diagnosis or assistance to the clinicians. So, you know, I think that there's a lot of variability out there. I think the best place for you to get a test is either through the public health infrastructure or at your specific doctor's office.
Okay, excellent. Carrie is asking, can you talk about the reporting requirements, please? There seemed to be confusion that there might be delays in reporting and a bolus of positive cases that don't correlate to increased frequency of virus in the community.
Yeah, so let me separate hospital lab or lab testing from reporting to the public health structure. So the hospital lab will test, and then you return the result. The result goes to the clinician and typically the patient, and that all happens in as real time as possible. At the same time, there's a reporting mechanism to public health, and that's going to depend on the lab's relationship to public health. So my lab, as an infectious disease diagnostic lab, is integrated directly to public health by computer. So what that means is every time I have a result come out, that result is automatically transmitted to LA County Public Health to be included in that day's data. Now, everybody doesn't have this connection. And so if the lab testing doesn't have that connection to public health, they may be faxing results over, and there may be delays then in that result landing at public health. And then remember, our public health labs are stretched to the max. Our public health infrastructure was not funded to take care of this type of crisis, right? We've been so good at limiting infectious diseases in the United States that public health funding for infectious disease, either contact tracing or even just taking in data, was completely cut over the last 50 to 60 years. So now suddenly we're thrown in a pandemic and public health has all of this data being sent at them. And remember, there has to be a person there, an expert there, to work their way through that data. And that's why you're seeing some of these delays. That's why suddenly public health will show up with a lot of data that maybe they've only accumulated over the last 2 weeks because either they were having infrastructure challenges of putting it together or delays in the reporting. Some areas that were doing testing didn't even know they had to report. And again, you're seeing COVID-19 labs pop up all over the place. And so it's really, really challenging, especially in a state public health laboratory, to get a hold of all of these different areas. But they're trying the best that they can. But Carrie, it's a great question. And it is why you see delays in the numbers coming out of public health. And that's the way this is going to be.
All right, fascinating stuff here. We've got time for 1 or 2 more questions. I'm seeing one from Lillian. Are we exploring any testing changes in the future to increase throughput and or faster TAT?
Yeah, Lillian, that's a fantastic question. And the answer is yes. So clearly the infrastructure in place, which is this sort of centralized testing PCR format, can't support all of the testing across the country. So one way that testing is being expanded is, of course, by antigen testing. This is like a rapid flu test, but a rapid COVID test, which are best used in areas that don't have access to PCR. So underserved areas across this country that are waiting 7 to 14 days to be able to test symptomatic patients, because a positive result is trustworthy on that. And remember, this is an area where nobody's able to get any testing. In addition, at UCLA, we've put forth a plan to increase testing by a method called sequencing. And sequencing is really where you can look at the nucleic acid of 10,000 to 100,000 samples all at one time. And so this is this concept of being able to by log fold increase the amount of testing that we're doing, and potentially for things like asymptomatic screening of businesses, asymptomatic screening of schools and campuses. To really support those settings, we need to be able to do way more tests than we can do now. And those testing platforms are just coming online. We're working on bringing one online at UCLA campus. And so hopefully we'll see that over the course of the next 4 to 6 months.
Excellent. Thank you so much, Dr. Garner, for taking the time to share with all of us in layman's terms. This has been so lovely. I want to be respectful of everyone's time. It is now 1 o'clock. So I'm going to stop the recording and the meeting at this point. Thank you again, Dr. Omai, for being here and for sharing with all of us.
For sure. I want to thank you all for attending. If you do have further questions, I'm more than happy to address them. If you want to send me an email, my email is ogarner, O-G-A-R-N-E-R, @mednet.ucla.edu. I'm more than happy to take your questions. I know an hour is not a ton of time to be able to discuss this topic, so feel free to email me, and thank you all again for calling in today.
And thank you all for being a part of our very first Staff Assembly Learn at Lunch of the 2021 year. See you next time.